So, welcome to the Protist's third set, part one lecture. So, I told you, this is a big group. There's just a lot of content to cover here, which is why I've chunked this down into three components. Don't try to tackle this all in one shot. I mean, it's just way too much to try to absorb. You need to do it in small, incremental bites. And that's, again, why I chunked it up. Do part one, look at that supergroup. Do part two, look at those supergroups, and then get into part three here and tackle these supergroups. It's a pretty extensive amount of content, so make sure you're processing it in small amounts. Otherwise, it's going to get overwhelming. So in the big picture, when we've worked through this at this point, we've already taken care of excavata. So we took care of that supergroup. Again, can you tell me what is the key feature to be in excavata? Within excavata, what's the key feature to be in the subgroups, diplomads, parabasalids, or euglenozoas? If you can't tell me, take a moment, pause, go back, look it up, just do a 2-minute review. And every time you do that review, that's going to sink in. And eventually you're going to go, yeah, I know it. I don't need to review anymore. I got this. We talked about chromoalveolata. Now that was a big group. Had two subcategories here. You had the Straminopilia alveolata. Again, what is the feature to go in chromoalveolata? What are the features for alveolata or feature? And then what's the key thing for Straminopilia? Once you're in alveolata, we had three subcategories. What are the features there? And do the same thing for Straminopilia. What are the three features there? Or the features for each of those three subcategories of Straminopilia. And then in the last part, part two, we took care of Archaeoplastida. What is the big characteristic here? What do you need to be in that particular group? And in Archaeoplastida, we talked about Rhodophyta, and then we kind of grouped Chlorophyta and Charophyta together under green algae. And then what is it to be a Chlorophyta versus a Charophyta? And land plants, don't worry about. In this unit, we will tackle land plants separately. Okay, those are going to be tackled as their own kingdom, their own group. All right, so now we still have three to go. We have Rhizaria, Amoebozoa, and Obstacanta. But the good news, these are the smaller ones. So we're going to tackle Rhizaria first and take a look at this supergroup and figure out what is unique about Rhizaria to put organisms into that supergroup. And then there are three subcategories: Radiolaria, Foraminiferans, and Cercozoans. Okay, so let's check this out. Again, here's the supergroup Rhizaria, and let me get rid of the hashtag and replace that with the red star. I want you guys to figure out and remember, okay, what is the key feature for an organism to be classified under supergroup Rhizaria? What makes Rhizaria different than the other supergroups we've looked at? And our big key feature here, excuse me, to be in Rhizaria, the organisms will use these things called pseudopods for locomotion. So pseudopod means false foot. It's a fake foot. What we're looking at here with this thing here called the amoeba is kind of an extension of the organism's body. This thing here, this is a pseudopod. So is this one. So are these. So amoebas are literally blobs that can extend part of their body out. Now, everybody who is in Rhizaria has these little pseudopods that they can extend out. They push them out, pull them back in, push out the other side of their body, pull back in. It enables them to move. Now, I've mentioned amoeba. That's actually in a different supergroup. They also have pseudopods. but the pseudopods are going to be different, and I'll get to that when we go into the amoebozoas in a little bit here. Okay, so let's stay on Rhizaria. Key feature, pseudopods for locomotion, 3 groups, Radiolaria, Foraminifera, and Cercozoa. So those are the 3 subgroups within the supergroup Rhizaria. Okay, so let's tackle the Radiolarians first. What is it about these things that we want to know and remember? Okay, so they have pseudopods. They use the pseudopods to eat, notice little quotes, their prey. So, oh, let's get spelling. All right, so hopefully this term phagocytosis may sound familiar. It's when a cell grabs something big, wraps around it, and pulls it in. Well, that's what Radiolarians do to their prey organisms, to the prey species. So they're eating tiny microorganisms, but they're doing it through phagocytosis. And again, it's a pseudopod that enables them to eat these things. Another big feature is that Radiolarians have these, we call them tests or shells that actually wrap around the Radiolarian and act as a form of protection. The shells are made of silica. So they're hard, they're durable, they're very solid structures, and then the pseudopods poke out of those shells to grab food and pull it back in. Now, Radiolarians are exclusively marine species. They're part of the marine plankton load. Officially, they are part of what we call the zooplankton. So this would classify these guys as omnivores. They eat stuff. They're microscopic, but they still eat other things that are floating around and live within a marine environment. So this is not the base of the food chain. It's one level above the base of the food chain. So pretty simple, pretty, again, microscopic organisms, but a very important link in the aquatic food chain ecosystem. You start at the base with phytoplankton, producers. So here's a big hint. Okay, let me red star this one. I want you guys to think, okay, big hint here. What organisms are, I'm supposed to say phytoplankton? We've talked about several. I'm supposed to say phytoplankton. I know, I'm sorry, my writing is terrible when I'm trying to use a mouse here. So which organisms are phytoplankton? The Radiolarians eat them. Radiolarians are the second link in the food chain in the aquatic ecosystem. Okay, so again, the big star hint should tell you that's an important thing. Go back to those sticky notes you should have been putting in your, or sticky tabs you should have been putting in your notes from the earlier part 1, part 2 lectures, and look up phytoplankton, build that food chain, start building it. Who fits at the plankton level, the phytoplankton level, and now Radiolarians come in at the first consumer level within the food chain. So, all right, so that's one of our subgroups of Rhizaria. The second subgroup are these things called Forams, or Foraminiferans for the official terminology. So Foraminiferans are also going to be part of the marine plankton load. So these guys will also play a role in the marine plankton. Now, these tend to be more at the producer level, at the phytoplankton level. And the reason they can go at the phytoplankton level is they have this endosymbiotic relationship with algae. So different algae will actually live inside of the forames and have a symbiotic or endosymbiotic relationship. And algae gives food to the forame. The forame then uses that energy and grows and does its thing. So it's kind of a weird dynamic. We would officially put these guys at phytoplankton because they contain the algae inside of them to be producers, but the forame themselves are not truly the producer. So classification gets weird. It's just goofy. It's not cut and dry. There's always gray areas and variations within it. So, but typically we would call forames phytoplankton because they run photosynthesis unofficially. They have tests. The tests are actually multi-chambered. Made of silica as well. So hard, durable, resilient. When the forame dies, the tests still remain because they're these solid, durable structures. And one of the really, so this is their ecological role. Ecologically, they're part of the plankton load. Where do humans use these things? Well, our value to them is that they are considered index fossils for the presence of oil. So when we go to the ocean and decide to drill the ocean floor for oil, often a geologist will take a core sample and they will, so if there's the bottom of the ocean, they will drill down into the ocean, pluck out a tube of sediment, pull it up, and look for forames. Now, if they find a bunch of forames in here, that is an indication there's a good chance oil is present. It doesn't guarantee it. Nothing's an absolute. But it says, okay, if forames are present in this concentration, that part of the ocean bed has been in existence for hundreds of millions of years, which is what it takes for oil to form. So oil and gas and coal, hundreds of millions of years for that stuff to develop and form. That's why we look for fossil evidence or forames, things like that, to indicate this stuff's been around for hundreds of millions of years. Otherwise, if you're looking at part of the ocean that's only 80 million years old, oil can't form in 80 million years. Thus why we call it a non-renewable resource. So, all right, now one of the really cool things about forames is if they build up in large, keep adding larges to this statement, large levels, they form this white, chalky sediment. So the White Cliffs of Dover, these cliffs are hundreds of feet tall, are white because there is such a massive accumulation of forames. Millions of years of these things dying and packing together and packing together and building up and building up and building up have led to the development and formation of these white cliffs. Okay, so imagine how many microscopic organisms it took to build a cliff hundreds of feet tall and give it that white appearance. So, okay, so I am going to pause here. I'm trying to break these lectures up at about 15-minute intervals and pause here. And then when we come back, we will tackle the cercozoas and that'll wrap up Rhizaria. So these last three supergroups will go much faster because they don't have as much diversity in them that we're going to be discussing during the Protist lecture. Okay, so let me pause here and then jump into part 2 of the third set of Protist lecture.